Hello everyone, my name is Hamzah. Today I would like to present our research. The topic is predicting the molecular structure relationship and the biological activity of DPP-4 inhibitor using deep neural network with CatBoost method as feature selection. I was collaborate with Mr. Alhadi Bustama, Mr. Aryan War, and Mrs. Devi Sarwinda. We are from Universitas Indonesia. Here the outline of my presentation. There are introduction, methodology, results, conclusion, and acknowledgement. Let's start the presentation. Diabetes mellitus type 2 is a chronic disease that often affects adults. In type 2 diabetes, the body cells become less sensitive to the hormone insulin. Insulin is a hormone produced in the pancreas. This hormone functions to help the body cells take sugar from the blood and convert it into energy. The enzyme peptidase 4 or DPP-4 can catalyze a decrease in the incretin peptide hormone, which results in a decrease in insulin synthesis. DPP-4 inhibitors are promising drugs for type 2 diabetes mellitus because they are one of the oral hypoglycemic classes that can block the action of the DPP-4 enzyme. The way DPP-4 inhibitors work is to increase incretin levels by inhibiting the action of the GLP-1 and GIP hormones to release glucagon, which in turn can increase insulin secretion and lower blood glucose levels. However, the DPP-4 inhibitors marketed today still cause some adverse effects such as nasopharyngitis, nausea, headache, skin reaction, heart failure, and hypersensitivity. So it's still expected that a new DPP-4 inhibitor with less side effects. Quantitative structure activity relationship or QSAR is a method to find the relationship between molecular structure and the value of biological activity. The QSAR method consists of QSAR classification and QSAR regression, where the QSAR classification of the target data is categorical, for example active or inactive compounds, while QSAR regression is to predict the value of biological activity from a continuous value molecular structure. How to predict the molecular structure of the DPP-4 inhibitor into active or inactive compounds? The method that is used to predict the DPP-4 inhibitor based on the relationship between the molecular structure and the value of biological activity has two stages. That is, the feature extraction method, and the second, the machine learning method. The feature extraction method is used as an initial step in building a classification model to represent the molecular structure of the DPP-4 inhibitor as a feature vector that will be used for the machine learning method at next stage. In the second stage, machine learning methods have developed as a method for predicting active and inactive compounds of DPP-4 inhibitors, one of which is the deep neural network method known as a machine learning method that is able to handle high-dimensional data. Vector extraction using the fingerprint method always produces high-dimensional dataset, so a method is needed to make machine learning methods work effectively and efficiently. Feature selection is one of the important steps for processing and analyzing machine learning methods effectively. Based on this background, this research will predict the DPP-4 inhibitor into active or inactive compounds using the circular fingerprint topology as a feature extraction method and the deep neural network as a machine learning method combined with the CatBoost method as a feature selection method. It's hoped that this research will be able to help someone who works in the pharmaceutical field to find compounds that can be used as drug targets for type 2 diabetes mellitus. This slide describes the methodology of this research, where inhibitor data is obtained from the online database site KBL. Then, preprocessing is carried out with several specified criteria. After that, feature extraction using the EGFP and FGFP methods, then the oversampling technique is used to overcome the imbalance of the dataset. Furthermore, the CatBoost method is proposed as a feature selection method. In the next stage, it's to learn the DNN model and evaluate the performance of deep neural network method. Feature extraction. In the first stage, the feature extraction method of extended connectivity fingerprint and functional class fingerprint is used as a feature extraction method. There are several parameters in shaping the molecular structure into numerical form, namely the selected diameters 4 and 6, and the resulting dimension are more than 1000 features. Deep neural network is method to predict the molecular structure into active or inactive compounds. The results of feature extraction in the previous stage are used as input factors in the machine learning stage. The deep learning method is used as a machine learning method in predicting active and inactive compounds of DPP-4 inhibitors. There are several hyperparameters defined in building the DNN architecture, including the activation function, initial weight, number of neurons for each hidden layer, optimizer, and regularization techniques. This hyperparameter can be seen in this slide. To measure the performance of deep neural network method in predicting DPP-4 inhibitors, the study used 4 parameters such as sensitivity, specificity, accuracy, and MCC score. This slide explains experimental results of this research. NIME software is used to carry out the pre-processing stage of data, such as eliminating missing values, removing salt, and etc. The target of DPP-4 inhibitor is determined based on IC50 values by defining IC50 values below 550 nm as active compounds, values above 500 nm as inactive compounds, and IC50 values in the range of 50 until 500 nm as the gray areas and are excluded from the dataset. From this process, more than 2000 DPP-4 inhibitor data were generated. In the salt removal step, it allows the molecular structures to map to the same structure with the same or different labels, so that the molecular structures needs to be removed in the study. Figure 7 describes the number of feature selection using CatBoost method. The CatBoost method is used as a method of selecting features from the extraction of EGFP and FGFP features. The author assumed that there are features that do not represent molecular structures well, so they need to be removed from the dataset. Figure 7 is the number of features based on the proportion of feature importance resulting from the CatBoost method. This is the performance of the DNN model based on MCC value. In this study, the MCC value is a more representative value when compared to the accuracy in evaluating deep neural network methods. The highest DNN performance was obtained when using EGFP6 as a feature extraction method with a feature selection proportion of 90%. From this figure, it can be seen that the feature selection in the EGFP method can improve the DNN's performance when compared to using all the features. However, the FGFP feature extraction method has not been able to improve the DNN's performance. Another advantage of using feature selection is that it reduces the compute time during the learning process and the prediction time. This is the base model of the DNN method to predicting the molecular structure into active or inactive compounds. The base model obtained from this study is the DNN model combined with EGFP6 as a feature extraction method using the feature selection proportion of the CatBoost method of 90% with sensitivity, specificity, accuracy, and MCC values as shown in the table. There are 2 points that can be concluded. The first, feature extraction using EGFP and FGFP methods are able to represent the DPP-4 inhibitor's molecular structure. It's also noted that the DNA method used to predict active compounds and inactive compounds, DPP-4 inhibitors are able to produce values of sensitivity, specificity, accuracy, and MCC score above 0.75. The second, important features recommended by the CatBoost method can improve the DNA model performance combined with EGFP feature extraction. The DNA model in EGFP6 and using more than 280 features obtain the highest MCC value of 0.810 and the values of sensitivity, specificity, and accuracy respectively are this value. This research is funded by Thesis Magister Grant 2020 from Kementerian Riset dan Teknologi, Badan Riset dan Inovasi Nasional. We also would like to thank all anonymous reviewers of this paper for their constructive comments and helpful suggestions. Okay, that's all my presentation. Thank you for a great attention and thank you for listening.